Good morning, everyone. Welcome to the RCN webinar. We're going to start at 8:03 as people trickle in here. Welcome to the webinar, everyone. I'm going to get started with some announcements before we begin. Today, we have two speakers. We have Marlene Wolfe, who's a postdoc at Stanford, and Hannah Greenwald, who is a PhD student at UC Berkeley. And this is the NSF-funded research coordination webinar for junior researchers. So these are for researchers who are PhD students, postdocs, junior faculty who in all cases switched what they were working on to devote their effort and provide service to the pandemic. So we appreciate the work of them and everybody on this call, and we all understand that this type of research is unique. And as Yesa said in a workshop last week sponsored by the RCN, we might all need therapy after the pandemic. So the way this webinar will work is that each speaker gets 20 minutes. And you guys should all put your questions in the Q&A, and then there'll be a moderator who moderates the Q&A and asks those questions to the researchers, and they can, attendees can upvote questions that they would like to see answered. And before we begin, I also want to say that today's webinar represents the last before the summer hiatus of these webinars. And Jill has put together a survey for you to indicate some feedback on the webinars and what you would like to see next for the webinars. I saw an unhappy face from one participant that we're taking a short hiatus. We could start up again in the summer, depending on the feedback that we get in the survey. So Jill is putting the link into the chat, and then she'll also email it to you. It should take you like 2 minutes or 1 minute to fill out the survey to give us feedback on what you would like to see next with the webinars. So with that, I also want to say that today is Marlene's birthday. So her birthday present is that she gets to present at this webinar. So with that, I will hand off here to Marlene.
How lucky am I? I regret telling Allie that it was my birthday, but it is true. Good morning, everybody. I am excited to talk to you today about our work over sort of the entire pandemic, doing SARS-CoV-2 monitoring with solids, and particularly looking at some daily high throughput analysis of settled solids from publicly owned treatment works in Northern California. Allie Boehm, who you know well, and Krista Wigginton are the PIs on this work, and we have a large team of talented people who have made this possible, and you will see their names throughout the presentation on publications and at the end. So I'm really excited to share with you today. Thank you for coming. Make sure my transitions work. Okay. So wastewater monitoring has really come into its own during this pandemic. We have this entire seminar series dedicated to it, and we've discovered how useful it can really be. To sum it up, wastewater provides a composite biological sample of the community. And since SARS-CoV-2 is shed in stool, we can identify viral RNA in wastewater and then ideally infer something about the incidence of the disease in the population. It's been amazing to have such a talented and dedicated community of people who are working on developing these tools to help protect public health during a pandemic. And as Ali said, I know so many of us have transitioned our research, and I have been very proud to be a part of the community of people who are working and collaborating on this. Wastewater monitoring uses a single sample to provide information about a whole community. It's also likely that the information from that wastewater can provide an early warning, right? People may shed SARS-CoV-2 in their stool before they're symptomatic or get an individual test, and we can report results from a single wastewater test that represents a community much more quickly than clinical testing can be aggregated and reported for that same population. But I think that the true strength is that wastewater testing is robust to changes in testing infrastructure and test-seeking behaviors. Whether tests are available or not, or they are sought or not, the wastewater will provide a value that's associated with transmission and unbiased by the availability of that infrastructure and those behaviors. And you can see that in this graphic here, where each one of these purple lines represents a case. Some of these cases have symptoms shown by this little figure, and some of them don't. Some of them are tested, shown by this, and some of them are not. So some of these cases have symptoms and are tested shortly thereafter, but in many cases, someone may be asymptomatic, have symptoms but not get tested, or have no symptoms and not get tested. And those cases, which are all infected with SARS-CoV-2, don't get captured by traditional surveillance, but do show up in the wastewater. And we see not only that there was sort of an under-testing at the beginning of the pandemic, but that now as we reach periods of lower incidence, that resources are shifting away from clinical testing in a lot of cases. But we still have a need to know what's going on with COVID-19 in our communities. So if we really want to have a wastewater monitoring infrastructure that can actually realize these benefits and serve as an efficient platform to reliably monitor outbreaks non-intrusively for entire diverse communities, what attributes do our methods need to have? And I would suggest that they need to have the following attributes. They need to be sensitive, they need to be representative, they need to be scalable, and they need to be comparable. And so what I mean by that is, for sensitive, we want to be able to detect low concentrations of SARS-CoV-2 RNA that represent few cases in wastewater. They need to be highly associated with incidence rates in the sewer shed. They need to be scalable. They need to be adjustable to a high throughput format of some kind so that we can regularly produce a lot of data to share with stakeholders. And they need to be comparable. They should be able to be compared across methods, because we know that to have every lab run things in exactly the same way will not always happen, even if you're using a similar set of methods. And so to do all of these things together in one method, this is kind of a story about our journey with wastewater solids over the past year. So first, sensitivity. From previous work by our colleagues, including work from our PI, Krista Wigginton's lab, we know that enveloped viruses, such as SARS-CoV-2, partition into the solids in wastewater. So that means that a sample of solids will naturally have higher concentrations without any manipulation of the sample, because the SARS-CoV-2 viral particles are sticking to the solids. Solid samples are also relatively easy to obtain. Already settled solids can be efficiently sampled from a primary clarifier. So this location in the schematic of the wastewater treatment plant here. Some plants don't have a primary clarifier, or for some reason it's less preferable or accessible. And from those plants, the solids can be settled out of an influent sample using standardized methods. So overall, it's relatively simple to get solids from any type of plant that you might be working with. And when we started this workout at the beginning of the pandemic in the US in early 2020, we wanted to start by looking at what was the best place to find SARS-CoV-2 in the wastewater, and if looking at directly at the solids would be the best place to find SARS-CoV-2 RNA. So we did a study to compare solids and influent at 2 plants in the Bay Area, comparing direct extraction of solids using a couple different methods to a PEG precipitation method to concentrate SARS-CoV-2 in the influent. For the influent, which you can see kind of the process on the top of this figure, we clarified the raw sample, add PEG and bovine coronavirus as a recovery control, allowed the sample to incubate overnight, and then spun the sample down and did RNA extraction directly from the concentrated pellet. For the solids, it was a little bit more straightforward approach. We dewatered the sample to get a higher solids content, spiked it with bovine coronavirus as a recovery control, and then did direct extraction of the solids. For both of those, we used that RNA template in droplet digital PCR to identify the N1 and N2 targets for SARS-CoV-2, along with that spiked in recovery control, bovine coronavirus, and pepper mild model virus, which many of you are familiar with, is an endogenous pepper virus that's excreted in human feces and is present in high concentrations in wastewater. So can serve as another way of looking at both recovery and fecal content of the sample. And what we found is that we had higher rates of detection, so fewer non-detects in the solid samples, and that the SARS-CoV-2 targets were present in about 1,000 times higher concentrations in the solids, as we had expected. So it was great to see that play out in the data. The measurements were also consistent among biological replicates. This work has been published, and I've got the reference down here. And this represents obviously a comparison of the PEG method and solids. And since then, we've done several comparisons of liquids versus solids using samples from California and Michigan and assessing different approaches to influent methods. And we still see this consistently higher concentration present in the solids. And so we were able to confirm that using solids is a sensitive method of detecting SARS-CoV-2. So confident in the use of these solids methods, we analyzed samples from 7 publicly owned treatment works around the country in our lab at Stanford using those solids methods. And these plots show incidence rates in the sewer shed at the top, the daily values, and then the 7-day smoothed number of cases per population. And then on the bottom, we have the measurements of N1 and N2. In copies per gram. And what you can see is that the wastewater measurements do appear to follow the pattern of the incidence rates over time for each of the plants. When looking more specifically at the association between incidence rate and these wastewater measurements, we see that the relationship between those wastewater measurements and incidence rate is significant. These plots show incidence rate plotted against N1 measurements in solids for those POTWs that you just saw all of the overwhelming data for. And we looked at the Kendall's tau for each of these using a bootstrapping method to incorporate error in the wastewater measurements. This first plot shows the raw copies per gram measurements against incidence rate, and that relationship is significant. However, it's significantly strengthened in this middle plot where the measurements are normalized by PMMOV. And that is serving to account for variability in recovery and fecal strength from sample to sample. And then finally, in this last plot, we have the measurements that are scaled by a model that incorporates not just the PMMOV concentration, but also the total suspended solids of the wastewater and the estimated partitioning coefficients for SARS-CoV-2 and PMMOV. And so that is a scaling factor, which we are calling F here. That again falls from a mass balance model that relates the number of SARS-CoV-2 fecal shedders in a sewer shed to the concentration of SARS-CoV-2 RNA in settled solids. I'm not going to go into the details of the model right now because I want to focus on how we've scaled this work, but they're reported in this paper here. And what we see, kind of the punchline is that although accounting for all of these factors does improve the association compared to raw values, it doesn't significantly change the relationship from the results normalized by PMMOV. And so that means that doing the relatively simple task of normalizing by PMMOV is a straightforward way to improve the relationship and examine the association between incidence rate and measurements in wastewater. So looking at this evidence, we can see that this data, particularly when PMMOV measurements are utilized, is representative of cases in the sewer shed. So we were very happy with this, and we were excited to then have the opportunity to scale up our work with wastewater solids to a more high frequency, quick turnaround monitoring project. So we enrolled 8 plants in Northern California between mid-November to December 2020. And each of those plants, which range in population from about 65,000 to 1.5 million people served, provide daily samples of solids that are brought to our industrial laboratory partner, Verily, by a courier. We then conduct analysis using methods that are adapted from those that we developed in our lab and used for the data that I just showed you at Stanford to allow for more automation and to be more high throughput, to allow us to actually process samples from all of these plants daily and return the results within 24 hours. The samples are also processed with 10 replicates, and we look at 3 SARS-CoV-2 targets, the N gene, the S gene, and ORF1A, and then those same recovery and endogenous controls that we were already using, bovine coronavirus and PMMOV. All of our protocols are available on protocols.io, and so I put QR codes here so you can follow them if you want to see the protocols themselves. For this high throughput version of the protocols using solids, first samples are centrifuged to dewater the solids. Solids are then resuspended in a DNA RNA shield at a concentration that we determined to minimize inhibition, and then spiked with the bovine coronavirus as the recovery control. RNA extraction is then performed using the Chemagic 360, and then followed by purification with a Zymo inhibitor removal kit. A Bravo liquid handler is used to automate many of these steps. It's not necessary, but it does help make the process go more quickly. And then finally, 2 ddPCR assays are run for each sample. 1 for the SARS-CoV-2 targets, and then 1 for our 2 high copy number controls, bovine coronavirus and PMMOV. Although this approach does require, as you can see, some specialized equipment, the use of solids means that it doesn't have any lengthy concentration steps, and it still achieves high sensitivity with rapid methods. And we've actually been doing some work to try to streamline the process and make it even more high throughput and simpler to perform at scale. So the data produced using these methods are posted to our website, which is wbe.stanford.edu, within 24 hours. And we communicate daily with our stakeholders, and we hold weekly meetings about these results. We've really placed a priority on developing these relationships with people who are providing our samples and using our data in order to make sure that what we produce is serving the communities that it represents. And we're very proud that Santa Clara County has started posting this data to their county website to share with the public in that way. Our website provides several different ways of visualizing the data. So overview plots like this one at the top, you can see we have 2 lines here right now for 2 of the genes and an S gene. And because we have daily measurements, we're able to take a trimmed average. And so what this shows here is a 5-day trimmed average where we drop the top and bottom values in that 5-day window and average the remaining 3. And that helps to acknowledge the variability that exists in environmental data and make trends more visible over time. And because we have daily data, we're able to do that. But we also have this drill down plot, for example, available. And that shows the raw data along with error bars that incorporate both technical and replicate error. And you can see the trimmed average line is also superimposed on there. And so having that overview plot makes it easier for stakeholders who are maybe less familiar with environmental data to digest what's happening over time in their community. So there are a number of different ways of visualizing, and you can kind of click around and play with the website to look at the different data in different ways. Over a period of 6 months, we've been able to utilize these methods of processing solids to deliver data directly to our stakeholders consistently within 24 hours. And so we can confirm, therefore, that this approach using solids is scalable. And now a little bit about the data that we've produced from this high throughput pilot. We've just completed an analysis of data looking at the time period from the start of the pilot, which was mid-November to mid-December, depending on the plant, through March 31st, 2021. And you can see here the data over time for each of the plants included in the study. There are 8 of them again. We did measure 3 genes, like I said, but they're so highly correlated that we're just only showing the results for the N gene here. In the top row is the N copies per gram in solids over time. The dots are daily measurements, and the line shows the 7-day trimmed average. So again, dropping the top and bottom values and averaging the remaining 5. The middle row shows N normalized by PMMOV over time, again, with individual measurements and a 7-day trimmed average. And then on the bottom, you can see the COVID-19 incidence rate for the sewer shed. And that's provided through data use agreements and shows the daily values and the 7-day smoothed average, not trimmed, for the incidence rate. And so visually, it's exciting to see that these do seem to line up really closely. But again, we want to look more directly at the associations. So here are plots showing the relationship between incidence rate and wastewater measurements for all of the plants, which are represented in different colors. On the left is the association between incidence rate and N copies per gram. So this follows the same kind of flow as the association plots that I showed you before. N over PMMOV is in the middle, and then N scaled by that F term that incorporates TSS and partitioning is on the right. To account for all of the variability in these wastewater measurements, I showed you the plot with all of the error bars. Kendall's tau and the regression coefficients were determined using 1,000 bootstrap resamplings to incorporate measurement errors, and then the median tau and regression coefficients are shown here. So for all of these scales, wastewater measurements and incidence rate were highly and significantly correlated. There's also a strong linear relationship between the log 10 incidence and log 10 wastewater values. For a 1 log 10 increase in N copies per gram, there's a 0.59 log 10 increase in N normalized in N copies per gram. And for both the normalized by PMMOV and scaled by F, for 1 log increase in those measurements, there's a 0.58 log 10 increase in incidence rates. We also looked at these results using a 7-day trimmed average for wastewater. So again, that trend line that you see on the plots. And while you can see that the data appears tighter on the plot, so like visually it's very pleasing, and we love these plots because they look like so beautiful with this straight line of data. Visually they look really nice, but the tau values are actually not significantly higher. So even looking at the daily data, we see these really high associations. And we can see both in these plots and the plots of wastewater values over time, that using a 7-day or a 5-day trimmed average is helpful for visualizing trends and reducing noise, but it's not actually statistically necessary to improve the relationship between the values. So again, for this high throughput analysis, we see that the wastewater data is representative of cases in the sewer shed. One more way of thinking about sensitivity is in terms of the lowest incidence rate of COVID-19 that can be detected by a method. So here you can see one of the plots from the previous page split out by plant. So this is the association between N copies per gram measurements and incidence rate. We use the linear regression models for each plant to predict the incidence rate when the wastewater solids contained our minimum detection limit. And here you can see the median predicted incidence rates for each of the plants. And so these values are for the plant, the data that's right above it, and then overall. And so overall, we estimate this approach gives us a minimum detectable incidence rate of 1.4 cases per 100,000 people. And this also aligns with our observed minimum incidence rate of 1.9 cases per 100,000 people at one of our POTWs. Over the duration of our study, we didn't see any non-detects in our samples, nor did we reach 0 incidents in any sewer shed. We are now approaching those levels, so it will be great to go back and do more analysis. We're still seeing how this plays out, but overall we can see the method is sensitive in terms of detecting very few cases. Another way of thinking about sensitivity, oh, sorry, wait. Okay. Finally, what about comparability across methods? So this plot shows the data from the initial methods development study that I showed in color, overlaid on the data from the SCAN pilot project in gray. The two data sets were generated in different labs using different methods during different phases of the pandemic and from different parts of the country, but both utilized solids. You can see in the far left plot showing the gene copies per gram and incidence rate, the colorful data set and the gray data set don't fall in the same cloud. But when the data are normalized by PMMOV or scaled by F, the data collapse and the clouds overlap, the slopes of the regression lines become more similar. That suggests that normalizing by PMMOV is an efficient way to make the results comparable across methods. And we believe this is primarily because PMMOV is adjusting for recovery. The median recovery of bovine coronavirus was about 10 times higher in the high throughput methods than in the colorful data set here. And I'm just wrapping up, Allie, I know. So we know that this is comparable, and that's very exciting to see we have a way to make sure that these methods compare across plants. So overall, solids enable an efficient, scalable monitoring platform that these methods are sensitive, they're representative of cases in the sewer shed, they're scalable, and they are comparable across slight differences in approach and users. And so we're excited to see kind of where solids take us next. I'm not going to get into this because I'm out of time, but sort of our next up is looking at variants of concern in wastewater. And so we have, several assays that we've deployed that have effectively tracked VOCs over time. So hopefully we'll have the chance to talk more about that in the future. But I just want to thank the incredible team that has contributed to this, because it's been a year of incredible effort by a lot of people in a lot of different places. So yeah, congratulations to our team on producing all of this data that I was able to share with you today. And I'm happy to take questions.
Great. Thank you, Marlene, and happy birthday. And thanks for
Thank you.
being willing to present on your birthday. So we have lots of questions here, and we're a little bit short on time. So, but I believe we had talked about in the pre-meeting, if we don't get to some of these questions, maybe Marlene could jot some answers in the
chat box. Yeah, and I put my email
well.
address here, so feel free to reach out to me too.
Great. So the first question is the possibility for false positive, and I'll insert this myself. I'm assuming false positive in this case, meaning a detection of an RNA signal that does not correlate with a case when the prevalence goes lower. So do you know how long this RNA signal could come from? And I guess this would correlate both to the transport of the solids through the sewage system and collection and persistence of the RNA. Have you all done any work on that?
Yeah, that's a great question. Yeah, I see sort of two things that contribute to that. One is thinking about transport of the virus through the system, right? And then the other is fecal shedding. And I think that in terms of transport of the virus in the wastewater system, we expect that the transport times between somebody's toilet and then the sample that we take are relatively short, less than 24 hours. And we don't expect to see persistence of signal in wastewater. I'm not aware of any studies, and it's challenging to think about an approach that would allow us to really see whether there is residual SARS-CoV-2 RNA that exists in the system that may then kind of give us a false positive later down the line. It seems very unlikely to me, particularly because that persistence would happen most likely at such low levels that it would be below our detection limit. And so that is sort of, that piece of it is less of a concern to me. I think something that we really don't know that much about right now, and where, lots of people are working on generating more data to help answer this question, is kind of what is the pattern of fecal shedding by people who are infected. And we know that sometimes that can last for a long time post the person being diagnosed. And I think that's one of the ways in which we think about the variability of these measurements, is we know that there is going to be noise in the data over time. And that while we see the best association between incidence rate, so daily incidence rate, daily new cases per population, most likely because people are shedding RNA in much higher concentrations at the beginning of their course of disease than later on, it is absolutely possible to have a super shedder or somebody who is shedding for a really long time contributing to the system. And that's why we think there's, this daily data and our ability to create trimmed averages helps us to kind of remove things that are actually outliers and don't really kind of tell us something about the trends over time. But this is a new area that we're getting into right now as we start to hit, 0 cases in some places. And as we start to get into the range of non-detect soon. And so I think we're going to have a lot more to figure out as it comes to that low incidence periods.
Great. And there's several questions in here and we're a little bit out of time. I'm going to ask if you can give a quick response, maybe just to try and slide one more in. Do you think solids represent a bit of a delay in incidence and sort of the relationship between this idea of early warning?
Yeah, I don't see any reason that solids would represent a delay in incidence compared to clinical data or compared to liquid wastewater. The samples are if they're from the primary clarifier, the residence time there is less than 24 hours. So it's a sample from that day. And if we're looking at settled solids out of influent, again, it's representative from that day. And we're doing that at one of the plants in our pilot, actually, they don't have a primary clarifier and we're settling solids out of the influent. And so I don't think there's any reason to suggest that there would be a delay compared to other approaches to looking at wastewater.
Great. Thank you so much. Happy birthday again. And there's a bunch of questions.
I'll answer more questions. I'll type in the chat. Yeah.
And so thank you everyone for your questions. And sorry we weren't able to get to all of them. So with that, I'll ask Hannah to share her screen and introduce Hannah Greenwald from UC Berkeley.
Hi, thank you. And let's see. Yeah,
that looks good.
Great. Thank you very much for the introduction. Thank you, Marlene, for that fantastic presentation. And happy birthday. I'm Hannah Greenwald. I'm a PhD student at UC Berkeley in the lab of Professor Carr Nelson, and I'm here presenting today on behalf of our amazing and extensive team that's been working on monitoring wastewater for SARS-CoV-2 throughout the pandemic. So today, my presentation will consist of 2 main parts. The first being I'll kind of give the backstory of Berkeley's regional monitoring effort, which we've titled COVID Web. I'll go a little bit into our methods optimization that got us to the methods that we use today. And I'll talk a little bit about our current data sharing. And then in the second part of the presentation, I'll highlight just a few of our research findings, mainly from a recent preprint that we put out that I co-first authored with Lauren Kennedy, including spatial and temporal trends of wastewater and clinical data, normalization biomarkers, and then I'll talk a little bit about our current projects. And lastly, I'll end with acknowledgements and contact information in case you have more questions. So regional monitoring effort, COVID Web. COVID Web stands for Wastewater Epidemiology for the Bay Area. And here on the right, you can kind of see a lot of our sampling locations dotted around the Bay Area. It consists of 38 sampling sites, which we sample 1 to 4 times per week. And this involves coordination between 18 different wastewater agencies and 5 county public health agencies. And we've been sampling locations across campus, as well as high-risk facilities, such as skilled nursing facilities, prisons, and locations at the sub-sewer shed level, as well as wastewater treatment plant influent. And all of these samples are processed in our pop-up laboratory, which we founded in October 2020, which has been slowly ramping up since then. And that's kind of one arm of COVID Web. The other arm has been our research arm. We've been conducting research on this topic since April of 2020, And we've had a few publications and protocols come out of it. And so this diagram kind of just shows our sampling, testing, and data analysis workflow. So like I said, we're sampling at various scales. All of our samples right now are 24-hour composite samples, and those wastewater samples of raw wastewater go straight into sample tubes that already contain preservative before they're shipped to us to help preserve them during transit. And I'll go more into the 4S method that allows us to do that in a minute. For testing, we're running most of our samples in biological replicate, and they go through the extraction process, and then we run RT-qPCR in technical triplicate. And right now, this diagram is a little bit outdated. We run SARS-CoV-2 N1 at 2 dilutions to test for inhibition, and then we run PMMOV and bovine coronavirus on every sample as well, because we've recently started multiplexing those assays. And then it goes through our custom Python data analysis pipeline. So the 4S method is something that we developed earlier on in the pandemic with some of our collaborators from different departments across campus. 4S stands for sewage, salt, silica, and SARS-CoV-2. And what really prompted us to do this is we had been testing some other kit-based methods, and with all of the back orders and supply chain issues, we really wanted a kit-free method with a fast turnaround time that could allow us to do high throughput sampling that we knew would be necessary. So the way this 4S method works is we have these tubes preloaded with salt and buffer, add the wastewater to them. Once they get to the lab, they go through a heat sterilization step. We filter out solids and then precipitate RNA with ethanol, and then we run that through a silica column. And then our template, again, is run on RT-qPCR. And the 4S method has been great for us, and we even have a Slack channel now on the WBE Slack, and other groups have started using it, which has been really great. Now, something that I took a big role in is optimizing our RT-qPCR and making sure it's documented. So I put together a detailed guide for getting started with qPCR. If any of you are in the phase where you're still getting started or want a little bit more of an extensive protocol, I can definitely share that. And then we also have a protocols IO where we really just assembled a bunch of pre-existing protocols for a bunch of these assays that are relevant. And then with some adaptations for like more affordable master mix, et cetera. A main challenge that we had in doing this project is figuring out how we wanted to deal with inhibition. So for inhibition testing, we've gone through multiple iterations of how we want to address that. We started off using the spike and dilute method, which is where you spike in your standards, our N1 standard, into the template and dilute it down, and then look at the expected versus actual CQs you're getting to see if the dilutions are inhibited. And that was just so resource and time intensive. And then it also delays the turnaround time, because after that, you then need to rerun the dilution without the spike in. So we abandoned that kind of early on and tried using an internal positive control. We used VetMax Xeno and spiked that into all of our samples post-extraction and had that assay multiplexed with N1. And we're using that kind of as a screening tool to see if we got different CQs related to a baseline before we would then run dilutions. But we later found, which it is pretty well established, that different targets can experience inhibition differently. And the VetMax Xeno target was definitely experiencing inhibition differently than N1. It wasn't really representing what was going on based on the dilutions. So we have since moved on from that, and now we run all of our samples at a 1x and 5x dilution The only issue there being that sometimes those 5x dilutions dilute out all our signal. We've also attempted digital PCR and digital droplet PCR, but with limited success compared to other groups. And recently we've been multiplexing some assays to increase our throughput, and we've been exploring multiplexed assays for different variants. We've also gone down the road of genome sequencing, and you can see a screenshot from one of our publications in the top right, which was just really early proof of concept using probe capture enrichment. We're currently using Arctic and NovaSeq, and we've had variable success, which has been sample dependent. You can see in the bottom right here, we're showing on the x-axis CQ value, and on the y-axis, the percent of the genome with coverage greater than 10x. So as you can see, most of the samples we've been running at this point have CQ values over 30, but the higher concentration samples tend to have more coverage, but there's still a lot of noise in here. We think that's due to degradation and possibly inhibition. We also have a dashboard now for data sharing. It is not publicly available. It is available to everyone on our project team, our public health partners, of course, and then also the wastewater utilities to be able to view the data. And it's really cool. There's multiple ways of viewing it. You can use the drop-down menu to see just N1 concentrations, PMMOV, N1 normalized to PMMOV, and you can see the different locations stacked or separate. And it's very fun to play around in and cool to see different ways to visualize the data and also to get that data to our public health partners. We also have a public Berkeley campus dashboard for our campus sampling efforts. This can just be found on the Berkeley website. It has a lot more limited information. It's mainly presence absence at our sampling sites, but it is available to the public. Our campus has decided to reduce clinical nasal swab testing moving forward, and wastewater sampling is to be determined if it will continue on past the summer. So in the second part, I want to highlight some research findings, again, from this preprint that recently came out. So we were testing wastewater from April to September of 2020. And here you can see on the right our results of the temporal trends. In the top, it's N1 and gene copies per mil. The middle panel is N1 normalized to crass phage. And in the bottom, it's daily new cases per 1,000 people. So that's our clinical results. And all of the lines are Loess smooth lines with a systematically chosen bandwidth, which we go into a little more detail on why we selected that and how we selected it in the manuscript. And something that really stands out with these trends is the yellow line, which is San Quentin Prison. We were able to capture the back end of a really big outbreak there where almost 50% of the population had tested positive for COVID-19, and it led to the highest SARS-CoV-2 concentration that we observed during the time period. You can also see that there's a general agreement of wastewater SARS-CoV-2 signal with daily new COVID-19 cases per population. I'll go a little bit more into the correlation in a minute. I did want to call out our normalization biomarker assessment. So what we've been calling normalization biomarkers are endogenous controls that are found in the wastewater that we can use to adjust SARS-CoV-2 concentrations for varying fecal content due to dilution of wastewater, maybe due to precipitation or due to sampling biases, et cetera. And previously there have been proposed biomarkers that are chemical, such as caffeine or total suspended solids. And more recently, pepper-mild model virus, or PMMOV, has been really commonly used in these studies. And we also wanted to explore some of the lesser used, but still ones that have been brought up, which like crass phage, Bacteroides, human 18S rRNA and flow normalization, where we did per capita flow normalization. So you multiply the concentration by flow and divide by the population that's contributing. And let's see. So for a biomarker to indicate fecal content, we wanted it to be consistently shed in feces across locations and seasons. And we had no precipitation during the study period, so we were really looking for minimal variation across both space and time of these biomarkers. So you can see on the left is crossphage, middle is PMMOV, and right is Bacteroides rRNA. And the x-axis of all these plots are the 6 locations that we sampled. And the y-axis is gene copies per person per day. So all of these values have been normalized to flow and population. So we're really looking for minimal variation across the locations, minimal variation over time. And the statistical differences across the locations are indicated by the asterisks. So you can see that crossphage across the locations had the least statistically different differences across locations. And then above each box are the geometric coefficients of variation, which is representing the temporal variation, and crossphage and PMMOV both varied less than Bacteroides rRNA. And you might have noticed that human 18S rRNA did not make it onto the previous slide. We actually abandoned studying 18S like halfway through the study just because the variability we were seeing was so high. It also experiences higher degradation than the other biomarkers and SARS-CoV-2. It's a common laboratory contaminant. So you can see in this plot on the right, 18S RNA is all the way over here on the right. And there was no statistical difference between the concentrations we were seeing in the samples and the negative controls. The samples concentrations in gene copies per mil are shown in blue, and the negative controls, the negative extraction controls, are shown in red. We were also seeing this really high variability where, again, the percents above the boxes for the samples are the geometric coefficients of variation, and for 18S RNA, it was 4,650%. And in a lot of the samples, we weren't even seeing 18S signal. So we don't think 18S is suitable as a normalization biomarker for these types of studies. But overall, our wastewater signal did have a moderate correlation to daily new COVID-19 cases. We also used Kendall's tau correlation. It's a ranked non-parametric correlation that's great for left-censored data. And our data is left-censored because I think about 25% of it was below the N1 detection limit. So when the data was unnormalized, there was a correlation of 0.43. When we normalized to flow rate, Bacteroides, and crossphage, the correlation remained moderate. But actually, when we normalized to PMMOV, our correlation dropped. It became a weak correlation of 0.18. And overall, this was surprising because, one, a lot of people were finding that PMMOV, and we were really expecting all of these biomarkers to increase the correlation or strengthen it. So we were surprised that none of them really improved the correlation, and PMMOV, the correlation actually became weaker. We also tried with our limited data to find a relationship between the clinical data and N1 normalized to 18S or total suspended solids, but did not find a significant correlation. So future outlook and current team projects. Right now, our team is currently trying to write up a pop-up lab blueprint for labs looking to get started in wastewater testing for future applications as well. We're trying to optimize a more swab protocol, which a lot of groups have already been working on, but we think this has a lot of utility, especially at the building scale, and we're really interested in it for our campus sampling, especially to save cost and be able to increase our sampling locations. We're also using our banked wastewater samples to test for other pathogens detectable in wastewater. such as enterovirus, norovirus, bocavirus, and polyomavirus. And this is both for epidemiological purposes as well as water reuse, which is what we were, what at least I was working on before the pandemic work. And we're really working to ramp up sequencing as a monitoring strategy. We are trying to start using and optimizing qPCR for the different variants. And we're still thinking about assessing the longevity of the pop-up lab and what its role is going to be at this phase of the pandemic and beyond. So with that, I just want to say thank you to our extensive team, our funding sources, all of the contributing public health and wastewater agencies that this work could not have been completed without. And I'll take questions, and then feel free to contact me or my co-first author on the manuscript, Lauren Kennedy, our project lead, Rose Cantor, or PI, Cara Nelson, with any further questions.
Great, thanks so much, Hannah. That was an awesome presentation, and we have a bunch of questions. If anyone else has questions, just as a reminder, please add them to the Q&A and we'll get through as many as we can. So the first one is about your method development. You mentioned that you've tried to do a switch to dPCR, ddPCR at some point without much success. Can you comment on sort of what the issues that you ran into were? And I imagine that other people switching platforms have had similar issues or trying to troubleshoot those, so.
Yeah, definitely. So, I mean, some of it I think could have been solved by the fact that we don't actually own a dPCR or a ddPCR instrument. So we don't have a lot of the institutional knowledge in using these pieces of equipment. Earlier in the pandemic, I went to a shared facility and tried to run ddPCR, and we're not sure if it's due to the 4S method or what was happening, but we were having trouble getting the droplets to form. We were having trouble with sensitivity. A lot of the samples that were amplifying with qPCR, we just weren't seeing signal with for ddPCR. And then more recently, we've partnered with Combinati and I can't remember who else, but we've been sending out samples to run on dPCR, like the microfluidic cells and ddPCR, and compared to qPCR, the results just aren't more sensitive like we were expecting, but we're still processing those results and moving through it. But yeah, it was surprising because I know a lot of groups did see increase in sensitivity with ddPCR.
Yeah, and if I can piggyback a quick question there, you mentioned that inhibition was an issue as you were developing your method, and now you're running dilutions. How frequently do you observe inhibition in the samples that you're testing? Is it like across the board, or is it certain sites more than others?
Yeah, I think it can definitely be variable across site, across day, just across that specific sample. But I'd say for the most part, now that we're running 1x and 5x dilutions, the concentrations that we've calculated in the 5x dilutions are generally higher than in the 1x, which we think indicates that there's some inhibition in the 1x sample. That's when we're not diluting out the signal at the 5x level. But yeah, it's pretty consistent where there's a little bit.
Great, thanks. All right, the next question is about your normalization to those different markers or different normalizing variables. So this is someone wondering how exactly you did your normalization to the different biomarkers. Can you comment on that?
Yeah, so we actually just divided the gene copies per mil of the N1 target by the gene copies per mil of PMMOV, crossphage, whatever biomarker. We had toyed with dividing them each by the median or different strategies, but we found that just dividing them directly was the most straightforward and didn't have a big impact to do these more complicated strategies. And then for flow normalization, we multiply the gene copies per mil by the flow and divide by population.
Great, thanks. Okay, so next question is about your clinical case data. So what measure of clinical cases did you use? Basically wondering what the source was. And do you know the expected lag time on that data when you're doing your correlations?
Yeah, so we're using daily new cases, and then they're population normalized. And all of these, we partnered with public health utility. We actually sometimes had to get the public health and wastewater people to talk so that we could geocode the data to the sewer shed. And they're generally, and they're moving averages, generally 7-day moving averages. For one location, we had a 14-day moving average. It's also all masked for privacy below 11 new cases per day. And then in terms of lag, we actually did explore lag analysis in the manuscript, and it was a more complicated topic than we expected because it's very location dependent. It depends on which time point you're using to like assigning to each data point, because sometimes we had episode date for the clinical data, which is the first date that the case was created. Sometimes it was the sample collection date. Sometimes it was the reporting date of that data. And likewise for wastewater data, you'd have sample collection date and you'd have reporting date technically, which includes the testing turnaround time. So for one of the locations, we actually had all three, and we were able to do a little bit more of an extensive analysis, but we had lag times ranging from 0 days to 3 weeks, depending on all of these factors. Also, normalization could even change the lags that we were seeing. But all in all, our analysis was a little more limited than some of the other studies that have come out because we did have weekly data, and daily data probably would have helped nail it down, but we still think it's really location dependent. So even if wastewater data is able to provide some early warning, nailing down a specific day is just going to vary so much by site.
Yeah, and I imagine, I think the person writing this question mentioned screening programs or incident symptomatic cases. With all the sampling locations that you guys had, especially across these different targeted facilities, there was likely, I imagine, a big difference in how the case data was being gathered depending on the specific location or facility, right? If it's just community surveillance, if you're symptomatic, go get tested, versus like a screening program, which is, yeah, a whole other complexity in that data. So, okay, we think we have a couple more questions and time for those. So with the temporal analysis, were you able to see impacts of certain public health measures that might have decreased concentrations or impacts of reopening when you were doing the temporal analysis? And then also with the spatial analysis, were you able to observe any higher concentrations in specific disadvantaged communities? I know you mentioned the really high concentrations at that prison outbreak, but did you notice anything else with your neighborhood data?
Yeah, so in terms of the temporal analysis, that was something we definitely wanted to explore, was looking at if we were seeing peaks like after the holidays and with different changes in shelter in place. And we even explored the option of using Google mobility data to kind of gauge people's actual movement and see if we saw any changes in the wastewater data. I don't think we really had the resolution to do that with our weekly sampling. Also in the Bay Area, we were pretty shut down for most of this period. So we weren't really able to dive into that analysis. In terms of spatially looking at disadvantaged communities, San Quentin prison had that massive outbreak where we were seeing the really high concentrations. In terms of the other locations we were sampling, we were able to see changes based on changes in the wastewater, like relative spatial differences that reflected the clinical cases. I'm not really sure about the the demographics of each of those areas, but we are exploring that further now as we get more into the sub-sewer shed level in our current work.
Great. Well, I think we are right on time, and I don't want us to go over by too much. So I think we'll close here. I want to say thank you so much to both Hannah and Marlene for presenting their work today. As Ali mentioned, we're taking a summer hiatus for the webinar series. I'm posting links to our feedback survey, and if you're not yet signed up on our listserv and want to get RCN emails, the link to sign up is also there. But again, we look forward to getting your feedback and then communicating with you all about our restart sometime over the summer or in the fall. So thank you all so much for joining us this morning, and thanks again to our presenters, and see you all again soon. Bye.